So welcome to the final session. So bear with us. You would really miss something if you book your train too early. So we start the session with the session is about data, different aspects of data, and we start with Catherine Aguilar from IPK, she would talk about rethinking non-genetic resources documentation in the age of data-driven science. So it's about bridging the gap between utilization and conservation.
Thank you. Good afternoon. I know we've been discussing significant advancements and cutting-edge technologies, but please bear with me as I take you a few steps back to the on-the-ground realities from the GMAP perspective, particularly on documentation and information management. Oh. It's okay.
You have to use the left or the right.
Okay.
Yeah.
Okay. All right. Thank you. So currently we have approximately 5.8 million accessions conserved in over 800 gene banks across 115 countries. Despite this extensive network, one of the criticisms that the global PTR conservation community always receives is the question, why do gene bank collections remain largely intact? And yesterday and even earlier, we heard from the private sector that one of the major hurdles is access. Access not only to the physical plant genetic material, but also to the information that accompanies these resources. Now, how do we bridge the gap from between conservation and utilization aside from all the legal frameworks? In plant genetic resources conservation and management, we are basically dealing with heterogeneous statuses. And you can see here the different data types that we are dealing with to describe a plant genetic resource accession. But of course, we'll be focusing on the phenotypic data or plant phenotyping. We've heard a lot about how complex. Okay, sorry, I couldn't cut loud. So we have been talking about the complexity of plant phenotyping, and I'll go through the challenges inherent to the field quickly. So basically, in a genetic resource center, we are dealing with different genetic profiles. Usually deal with this four, and each genetic profile requires different strategies. How you handle SSD lines is different when you handle heterogeneous accessions or within accession heterogeneity. And then, of course, we are dealing here with different crops, and each crop has their own diverse range of traits, different growth patterns, and we need to capture data in all developmental stages. We need to consider the different interactions, soil, microbial communities, light conditions, atmospheric conditions, and all the other environmental and management practices that we need to consider as well. And of course, phenotyping spans across different scales, from molecular, genetic expression and proteins, to the organ level, individual plant level, field scale assessments, and even to agroecological field assessments. And then we are working under different contexts. Each institution has different objectives, different projects, different infrastructure, different expertise. So here I'm showing the Phenosphere from IPK, and then some lemnotext communalizers. Then we have the usual traditional manual phenotyping that I believe most gene banks are still doing. As what we have already mentioned several times, there are different stakeholders involved and diverse data contributors, not only gene banks, not only plant breeders, researchers. But today's reality in gene banks is a mosaic of diverse practices. And you see that we are working using the crop-specific descriptors, but oftentimes we modify these descriptors to suit our needs. We have varied data collection methodologies. There are different data reporting and exchange formats. variable naming and semantics. One gene bank might have these names that the other gene bank may not be using. Disparate information management systems. So historically, gene banks or research institutions develop their information management systems in silos. And of course, gene banks are dealing with a multitude of historical data sets that are very difficult and tricky to standardize. Here I am just showing a snapshot from Pieruschka where we can see very old data sets. So you can see here as old as 1940s. So right now we are dealing with ambiguous IDs. Earlier this morning, we heard that one accession might have different identifiers. Of course, the challenge about data fragmentation. So you see, one must deal with the different databases. And most of the time, we have this project-based databases that after the projects would be decommissioned or archived. So you can no longer access the databases. And then some would still have their lab notebooks or data sheets or keep their data in their personal computer. Then we have this compromised and incomplete metadata sets. And with the advanced technologies right now, we have the challenge of dealing with high volume and very complex data sets, and the challenge of accessing this data set. So the question would be, how much of your data can be accessed by the users? Or if they can access the data, do they understand the data? So that is a challenge. So this is a very good example, which was actually raised again earlier this morning, that one accession distributed to different institutions, or probably even within the same institution, would have different codes, different identifiers, and it would be subjected to different experiments. And as you can see, no unique identifier, incomplete metadata, and highly fragmented data sets with varying structure, various formats. So the challenge is, how will you integrate these diverse data sets to accurately attribute them to the specific accession? But these challenges are not really unique to PGR. I believe many fields across life sciences also deals with these challenges. And so there's this global push for data fairness, making our data findable, accessible, interoperable, and reusable. So for our data to be findable, there must be unique and persistent identifiers. Example for that would be the DOI. So every accession must have unique identifiers. The experiment, the study must have their own unique identifier, even the equipment used. And the data must have rich metadata. There should be clear metadata data links, and of course, must be searchable. For you to access the data, it should be retrievable using open and standardized protocols. It should allow authorization when necessary, and there should be clear licensing rights. And I must stress that having our data in publications, even as supplemental tables, does not meet the requirements for full accessibility. Then for our data to become interoperable, we must make use of consistent vocabulary. So we have control vocabularies, ontologies. Our data must have formal, accessible, shared, and applicable language. And of course, the shared standardized formats. So we have crop ontology, and there are various initiatives on standardizing the semantics. And we have for phenotyping experiments, MyAP, which I believe Cyril will discuss in detail later on. But I must admit that even if these steps might be straightforward, many of the gene banks lack the expertise of carrying this out. I myself am not an information specialist, so I know very well the struggle of getting through all the different standards. And for our data to be reusable, there must be detailed information about the provenance, rich descriptions with accurate and relevant metadata, and again, meet the domain relevant standards. Of course, there have been several initiatives in pushing for fairness of phenotypic data. I just have here some. We have Germinate from James Hutton Institute, Bridge Portal from IPK, and then we have the Genetic and Genomic Information System and FITS, and of course, the emphasis. Currently, Eurisco is using this simple data model. So it's basically derived from minimal consensus. We must take note that Eurisco is working within a network of 43 countries with 400 institutes. And these institutes have varying capacities, varying resources. And so Eurisco, regardless of these limitations, is of course working towards complying with all the standards. So in a nutshell, what we need to do is first to implement unique identifiers, such as the DOIs. We can make use of the infrastructure of the PAN treaty. We can use it for free or assignment via Eurisco. You can ask your national focal points to request Eurisco to register the accessions through GLIS. And then we must adhere to the data standards, both the semantic data standards and structural data standards, as I've mentioned earlier. And of course, the implementation of best practices. Each GMAT or research institute must have their standard operating procedures for data collection and documentation. One must have quality assurance and validation, data management plans, especially for project-based data, and of course, training and capacity building. And finally, the implementation of technical standards. If you want to integrate different data sets like genomic data or various multi-omic data sets, and you have the example here for breeding API for Gabi. So within the context of Progress, we are working towards coming up with a proposal for a coherent guidelines building upon the established standards. So one of the standards would be myopy, but as I've mentioned, we are dealing with different data types. Each data type has their specific data standard. So we want to come up with a harmonized, coherent guidelines, and tentatively we call it as MyPGR and Eurisco as still the central hub of Progress. So way forward, again, regardless of institutional capacities, documentation is non-negotiable. And if you are overwhelmed looking at the standards, you can just start small, baby steps, pragmatic approach to data collection and management, and then plan for scalable growth later on. Foster the culture of fairness. And as always, we have to bear in mind ethical data stewardship. And training and support is very important, getting feedback loops, and like what we are doing now, collaborate across networks. And just a concluding thought, it's not just about having the best tools, it's about bridging the gaps in our data practices so that our data remains useful, not just for today, but for the future. And the challenge that we have now with millions of underutilized accessions or tasks is clear, turn potential into progress. That would be all. Thank you.
Thanks a lot, Catherine. So, okay, I'm pretty sure there will be lots of questions. But we are moving on now to the next speaker, which will be James Brett from the Earlham Institute, talking about tools to manage bio, phenotyping, and marker datasets. This is an experience from a Legume Generation project, so it's breeding of legumes in Europe. Yes, the stage is yours.
Thank you very much. There we go. So I'm just going to basically give a little overview about some of the tools that we use in Legume Generation to do our sort of data management for our phenotyping and field trial data, and I'll touch on market data sets as well at the end. So Legume Generation is a big European project. We have about 40 different partners, and these partners are basically split into 6 different packages. They all have their own individual crop that they're working on. And they're all running their own field trials, their own phenotyping, and their own genotyping analyses. So obviously this produces a lot of data. And my role in Legume Generation is basically creating a sort of knowledge center where we can, at the end, make all of this data accessible to breeders or anyone that's interested in it, and so that they can make more informed decisions on breeding programs and sort of accelerate that crop breeding development. However, producing all of this data creates this sort of problem of where do you store it, how do you store it, and how do you make this data accessible to people? Ooh.
Yes. I suppose.
Is it a TV?
I think it's a TV, but, oh. It's a TV.
What do you?
There we go. Okay. So how do we make this data available?
Watch.
So one of the sort of tools that have been developed at Earlham is grassroots. So I haven't developed this, but Simon Tyrrell has, who's the main developer of grassroots. And what this is it's basically this software that allows you to store all this field trial and phenotyping data using these FAIR principles. And I'll very briefly touch on what FAIR principles are, since we just had a nice description of them. Basically, it's data that's findable, so data that's described with this rich metadata. So it describes exactly what it is. And it's machine and human readable. Accessible, it's no good being able to find the data if you can't access it, which is something grassroots specifically deals with. And that this data is retrievable with these unique identifiers. Interoperable, so integrating different sets of data sets. So, for example, integrating your phenotype and your genotype data, and also integrating these data with different workflows of analyses. So maybe like running a GWAS or genomic selection. And reusable. So the metadata that describes your data should be so well defined that anyone can look at it and completely reproduce it, or integrate it with or sort of compare in a different sort of setting. So, for example, different environments. So one of the main services we're particularly interested in with grassroots is the field trial management. And so what we have with this is we have a standard template that we sort of dish out to all of the different partners. And this template has all the metadata already sort of predefined about the field trial layout, plot, row, etc. And then they can define their own phenotypes as well using sort of standard crop ontologies. Or we can make these custom ones that we'll then submit to crop ontology. And so this is basically to facilitate the publishing of data that is compliant with these sharing principles. We have a number of different programs that already use grassroots. You'll notice that they're all wheat, but that doesn't mean it's specifically for wheat. Just delivering sustainable wheat is quite big wheat project. But the idea of grassroots is that it's crop agnostic, so you can, in theory, use it for any crop you want, which is why we've decided to use it for legume generation. It's not specifically for wheat, don't worry. So the way that data is findable in grassroots is we have a number of different ways. So the first is a very nice little interactive map, so you can go on as a user, scroll through the map, see where your field trials are located, click on it, and it will give you the data associated with that. Or if you know more specifically what you want to look for, so you've got some keywords, there's a search text box. And this is just available on the web browser. You can say keyword and put the associated experiment and all the data and metadata associated with that. Or if you're more tech savvy, you can integrate this programmatically. So you can write a program and sort of submit these JSON requests in your program to pull this data from the data of the server. Accessibility. So once data is published, it's completely open access. So anyone can pull out data if they want. So nothing's locked behind any sort of paywall or anything. And again, all these data have these unique identifiers. So if you know what that is, you can find it, and then you've got the data. And interoperability. So grassroots can, it's got its own API that you can sort of interact with. And it also works with the BRAPI, so the breeding API. and this lets grassroots communicate with other services that also communicate through graphic to run sort of analyses that you want to do. Frictionless data, I'm not entirely sure what that is, since I'm not the lead developer on this, but I've been assured that it's positive. Can't tell you any more than that. But there's also CSV files, so that's like the standard column, comma separated file that you'd input into any sort of workflow. You can also open those in a spreadsheet, so if that's how you want to look at your data, that's easily doable. And reusability. So what we use is a set of standard ontologies, as I mentioned before. So the main one that we're using is crop ontology. So obviously we've got all these different partners with different crops that are doing their own phenotypes. So we are working towards using these crop ontologies to have a set list of phenotypes that, if you're talking about height, everyone's measuring height the same way. For example, stuff like that. So the crop ontologies can sometimes be quite restrictive. So grassroots allows for defining your own custom ontologies and then submitting that to crop ontologies so that it becomes a standard term that everyone can use. And then again, we have the same sort of standard ontologies for field trial data, plot, row, width, and things like that. That's all standard across every single field trial. And because if everyone measures their field trials in slightly different ways, it makes it really difficult to sort of compare between sites. And then finally, how we're sort of working with genetic data. So we can't use grassroots for that because it doesn't quite have the capacity to store millions of markers for 6 different crops and however many accessions. But we've been looking at 2 different softwares. So one's called Rebase and the other is 333. And both of these have been used in other projects to produce quite nice databases already that sort of houses same information. So Cassava base based on Rebase and Nopals, which is quite relevant to Legume Generation. So we know what it's going to use. And it stores all of this genetic data, and both of these also interact through the bracket. So the idea is to get the market database communicating with grassroots so that you can run these sort of downstream analyses. So it's like calculating loops and blues or running a genomic selection pipeline. And that's also done automatically in backend. So that's the idea. And then just like to acknowledge everyone that's involved. So Jose de Vega, who's the lead in the data management for Legume Generation. Simon Tyrrell, who's the main developer and all of his team. And then John and Rothenstedt and the University of Bristol that have sort of helped fund and keep grassroots going. Thank you.
So hello, thank you for the invitation and for the many teasers. So I'll do my best to show you how on for this infrastructure Elixir, with a collaboration with the CGIRs for many projects, we have built a series of tools of principle to enable data management of phenotypic data. So first, thanks to my colleague for introducing the FAIR principle. Therefore, I was right to skip those slides. As a reminder, what we want to deal with is a series of concept tools along the data life cycle with planning, it's not working, collecting, process, analyzing, preserving, sharing, and reusing, either for high throughput, highly automated platforms, or for more traditional phenotyping networks. And those platforms, those networks have to deal with different situations, therefore they have different answers. First, especially for highly automated platforms, we need dedicated information systems such as FIRS, which has been mentioned before. So it's for instrumented and complex experiments. It works. Yeah, not that easy, but it works. And as you can see on that part, it provides a lot of traceability, very detailed traceability. but so detailed that it is not always necessary, especially for traditional or hand measured data. And for that, we are seeing also in our communities simpler data sets, simpler phenotyping and data management using simple Excel files or CSV files, like what we have just seen in grassroots. And this is in our case, using the Phenotyping system. But in all scenarios, the first step is datasets that are organized for data acquisition, which is not the same as for data analysis. Therefore, there is a second step, which is about producing this kind of tables, which change something that is rather complex in a plain old table. And this leads us to the next step, processing the data, analyzing it, then preserving it in databases, and finally doing data publication, data paper, and scientific paper. In fact, it's way more complicated than that, because we are putting together several circuits like that from networks, from platforms, etc. But the idea is here. And what I'm going to quickly present you, and this is more cheat sheets that you will be able to review later, is some tools, some systems that can be used for data planning, collection, scripts and workflow, data repositories, including Euroscope, and data portals, because as you know, there are many data repositories, and we need to be able to share among all of those. Sorry, a little bit too quick. And since we have many tools and many repositories, we need something to link everything together. So this is why we've got those global data standards in the middle. So we have heard a lot about MyAPI, about BRAPI, about crop ontology. And as you will see, having standards on tool is nice, but without guidelines and training, it's not enough. So the plant data standards. The first category, and there are many, is about structured data standards. It's how you format or organize, of course, your data, but here you all need a lot of leadership, but also your metadata. In other words, the description, what are the information you need to gather to enable data reuse. So it's data models, template, it includes a list of phenotyping variables, list of biological material, and it is, to be efficient, built by biologists and computer scientists. And as you can see with those arrows, it embeds a list of possible values coming from semantic standards, so description of the data. It's the ontologies, and the main example is a crop ontology, as well as for biological material, of course, persistent unique identifiers. So it's the DOI system for the glaze, it's the MCPD and so on. And I will not go into too much detail about the technical data standards, but it's a way to make automatic use of the standardized data, such as MyAPI and crop ontology. So those semantic standards, I know that you have already embarked some of those information in some of your daily reports. That's great. Nonetheless, getting a little bit back on it. So the crop ontology approach is about building a triplet, trait without scale, because agreeing on trait name is absolutely not enough. For instance, if we've got here plant height, we know that we've got at least 4 ways to measure the plant height, or even 5, if we are dealing about high throughput, and a couple of units of measure, which means that for a single trait, rather simple one, we've got at least 11 phenotyping variables, which is quickly a lot. Therefore, we need systems to be able not only to formalize those variables, to describe them without any ambiguity. It's not a couple of fields, as you can see here with a little example. But also, we need to agree on either the reference ontology, so that's the crop ontology, or at least a model, a framework to use in your own data set. Indeed, contributing to the crop ontology is time consuming. We know that breeders can be rather creative with their variables. and we must leave them the possibility to advance with the science. Phenotype structure standards. So for phenotyping, it's minimum information about plant phenotyping experiments. Important, this is for phenotyping, but it is also embedded in the EBI databases, hence enabling interoperability with sequence data, with genetic variability data for now, and later we hope with metabolomics. So every information is available on myapi.org. I will just give you an overview. There are trainings everywhere, and especially one at the end of the year, if you are interested. MyAPI, it's only a specification. We have made our best effort to make it mostly biologist friendly, keeping the troubles for computer scientists. So a simple specification table in it, with definitions, with examples, metadata fields, and a data model that simply states that an investigational dataset has many studies or experiments, each study is having its biological material list, its observation variable list, etc. And therefore, since MyAPI is only a specification, it needs to get implemented, of course. And therefore, here comes the technical standards and the implementations in general of MyAPI. First, the databases, the data repositories. Ideally, the data standards should be mostly hidden from the users. The tools should take care of standardization. So, in fact, any breeding API compliant database is a MyAPI compliant database. So there is GenPIL, there is FIS, there is PIPA. There is a lot of databases such as BreedBase that have been mentioned just before from the CGIAR and so on. As you will see, there are also some generic data repositories that are broadly MyAPI compliant with Dataverse and Zenodo. There are also file systems. We want high-end approaches with databases, but also very simple, pragmatic approaches using file template. So there is an official MyAPI template available on GitHub. There is a minimum version of the MyAPI because it was a little bit frightening. So it will be available next week, I hope. From this, we have the project workflows. We have also ISATAB and AeroCreate file archive. Web services, the breeding API is a MyAPI implementation. It's a web service specification built by Elixir and Phasis, and especially the Excellence in Breeding platform, who has been paying full-time coordinator for 5 years, which is great. And finally, we've got an ontology on all implementation methods for computer science. Very quick, a single slide on tools for data collection, experimental information system. So, which, as I said, are mostly MyAPI broadly compliant for those I know which are in production. So, FIRS, PIPA. FIRS is an ontology-driven information system for plant phenomics, and it allows to integrate environmental data, phenotypic data, high throughput. It can be used also for hand phenotyping, classical phenotyping. It's always a nice and sympathetic tool. And a few weeks ago, somebody asked me how to build a Brapi database, and after 15 minutes, I thought, okay, stop, use that, fix, great. And he looked at it. Anyway, there are also, as I said, phenotyping network systems, so really file-based, simple, using BreedBase of Fairdown. In the agent project that we mentioned before, we have built a workflow using a simplified version of the MyAPI file template. This workflow will be published soon in the Zenodo agent project community. So I suggest that you keep on that one. Our idea will be first to publish it almost as it is. And after, we want to generalize it a little bit because by discussing with a lot of people, it drew really some attention. And in that, so we've got a central repository, Fairdown and a file validator for this file template based on instructions. Yeah, for the portable devices sensors, I just wanted to pick one. There are many, but I like this one. So the field book, which is BRAPI compliant, available on GitHub, totally open field, of course. It allows you using a smartphone or any Android device to go in the field, in the greenhouse, make measurement, load your experiment using BRAPI, and export what you have measured using either files or breeding API directly. For data repositories, the only thing I would like to stress here is that there are generalist data repositories, Zenodo, Dataverse that can publish and share any type of data and that are very interesting for phenomics dataset. And moreover, they are good because you can use general metadata, year, data type, author, the organism without any controlled vocabulary, keywords, very, very basic, just as any publication. But there are the possibility to add phenomic-specific metadata using the guidelines that you can see over there, and I will tell you right after where to find those guidelines, which is simply about adding 3 companion files with the biological material, the observation variable on the list of studies. Finally, since there are many data repositories, I will just quickly mention the FEDER data portal that indexes most of those data repositories, 30 today, 40 today, and provide a full text search and link back to the original database. Finally, guidelines on training, just 1 minute. First, I would like to underline the RDM kit, and I know that some of you at least heard about it. It's a Wikipedia-like knowledge base, it's free, it's open, it's maintained by a community of nearly 200 people today. It describes how to manage research outputs according to FAIR principles. And the entry point for us as plant researchers is this plant page. So it has been built within Elixir and with contribution from Emphasis. If Progress want to contribute, you are super welcome. And finally, trainings. So we have been training since 2018 or even 17 on a lot of the tools and standards that I've presented to you. There will be also some joint training with Emphasis, Elixir, Fenet, AgroServe organized, and there is in particular one from the 3rd to the 6th of December that I'm currently struggling to. And I hope this is the last acknowledgement slide in date, and that I forgotten nobody, but that's the work of Roland.
So the questions will be afterwards, and I'm very much looking forward to the last talk. So not the reason is not because it's the last talk, but because it's a really interesting application how to use metabolomics data. And so basically not just talking about data management, but an example how to use data and make predictions from metabolomics.
And reuse data.
And reuse data, and I did not mention your name. So very happy to have Yves Gibon from Bordeaux here.
Thank you, Roland, and thank you for staying for the last talk. Yeah, so just to mention that, so we have a platform, metabolomics platform, at the interface between that works at the interface between Phenom and MetaboWeb. So the title could have been Phenom when Phenom met MetaboWeb. So the title, by the way, I took it from this interesting opinion paper from 2016. So the author, in a unique, funny way, described new techniques arriving in the world of plant breeding. So you see QTEL mapping, later on GWAS, and more recently genomic selection. So you have a period of excitement and then the bitter reality. And the funny thing was he wrote somewhere in the paper that metabolomics never achieved the bandwagon status because considered too complicated and too expensive right from the beginning. It's a pity because there's lots of information in metabolism. And well, as you know, metabolism provides energy and bricks for biomass construction, defense, attraction, signaling, and many other things. There's also temporal information about the past, the present, and future. And I think since 2016, there has been lots of progress, and in particular, the combination of robotics and high-resolution mass spectrometry. On the one hand, we have some achievement regarding throughput and also the density. So with a higher resolution mass spectrometer, you have lots of information. You can go up to 20,000 variables that correspond to chunks of metabolites. So you see this picture on the right. So we try to depict the throughputs of each step in the metabolic pipeline. And you can see two major bottlenecks. So the sampling, of course, which is mostly manual, unfortunately. And it will be very interesting to try to improve it, to automate it, especially on phenotyping platforms. And the other one is the analysis. So it's a time an extract needs to pass through chromatography and mass spec. It's around 10 to 20 minutes. So if you run 24/7, you will have like 20,000 samples per year with one mass spec. So that's the throughput we have in Bordeaux. And now about the cost. So it's a rough estimation. Maybe I'm too optimistic. So the most expensive thing is the equipment, but it lasts for 10 years at least. And then you have maintenance costs, running costs, human resource. And so I estimate it costs around 200,000 euros to run just one mass spec. And so you divide by 20,000 and you end up with 10 euros per sample, which is not that much. And if you ask Google, Google will tell you it costs between 50 and 500. Okay, so now about the complexity. This is a quick pipeline of how the data is used and what it represents. So the data that comes out of the machines is quite big. It can be tens of megabytes to gigabytes per sample. So it's a lot. If you have 20,000 times 1 gigabyte, it's 20 terabytes a year. We have only 100 terabyte server, so well. But luckily, we can drop the data into a public European repository, Metabolite, and we have to add metadata. So I think this works. You can get the metadata if you provide a nice service. And so the data needs further to be further pre-processed before we can use it for statistics and other things. And here there is an increasing number of open source software that is really nice. For example, XCMS or MSDial, that does all these operations to render the data interoperable. And so a very important point in metabolomics, you need to include so-called quantity control samples or extracts. So every 10 or 20 measurements, you need to put them because you have to take into account the drift of the machine. So mass spectrometers are not stable over time. And so they are used to normalize the data to make it interoperable within an experiment, so even a peak with thousands of samples. And it could also work between experiments, but I doubt it will work if you want to integrate experiments and measurements on different platforms, different brands of equipment. Well, and then the data analysis, I mean, it's nothing special, multivariate analysis like principal component analysis, or supervised, and I wanted to focus on the top-down modeling because I think it's relevant for phenotyping. So we start from diversity panels, of course, the genotypes in intraspecies, but it can also be interspecies diversity. One big advantage of metabolomics is that many metabolites are shared between species, so that's an interesting feature. And so metabolic features will be the predictors. We don't need to know the nature of the metabolites actually here. And the traits to predict, as has been mentioned over the 2 days, performance traits or also robustness traits, whatever. And the modeling approaches used are also classical. So we use a lot multilinear modeling in with a package such as GLNet in R. And well, it works nicely. Of course, we need a supercalculator for that. And I'll put you 3 examples of successful predictions. So we could predict the relative growth rate of the fruits, whatever the stage of development. Model multi-species model. A grafting success in grape wine. So this is very strategic in Bordeaux, of course. And the most striking one was probably that we could predict the elevation at which plants grow in the Atacama Desert based on the metabolomics. And so, and from there, get some idea of what happened in metabolism to adapt to these harsh conditions. Back to plant phenotyping, this is something we wanted to do for years in Phenom, is to make 2 platforms work together and try to get some real prediction. So the idea is we would take samples from young plants in the greenhouse, perform metabolomics, and build a predictive model of yield in the field. So really separate the predictors from the target trait. And if that works, then we could later on try to identify the best variables, so what metabolites are behind, and then go into elucidation of mechanisms, for example. And so François already mentioned this experiment, a very nice experiment with these hybrids that were released since the 50s. And you see this nice linear increase in yield. And we wanted to see if something in metabolism would have been selected as well. And the result is quite interesting. So to get an idea, we built each time 100 models for each traits they had to propose to us. François, Claude, and co-workers. And then 80% of the data was used to construct the model and 20% to test it. And so you see the data is, the box plot show the dispersion of the R2, so correlation coefficient between predicted and actual data. So on the left, it's the traits that have been measured on the platform is pretty nice, but well, it's within the same experiment. More interesting is the year of release, a very good prediction, an R2 of 4.8. And we could further validate it by constructing the model with every year except one and check with this one. And you see there's a very good correlation as well. And finally, about the yield in the field, you see it works really well. Most of the predictions are above 0.75. So because there were different scenarios tested in the field, different field trials, with or without irrigation and so on. And you see it works very well. So to conclude, I would like to say metabolomics has progressed. And the problems that are left, I think it's mostly sampling, because it's destructive observation, and the interoperability. I mean, I heard several times metabolomics data have to be stored in databases and so on, but I mean, they have to be interoperable if you want to integrate them. And then the key point was to work on the trade-off between prediction value and costs. Cost decrease when you increase the throughput, as simple as that. And I think, I mean, we could further improve it and get down to less than €5 per sample. It's just a matter of throughput. And as I showed, metabolomics can sometimes provide very good predictions. So now, when to use predictive metabolomics? I don't think it makes sense to compete with genomic selection in major crops. So, well, I don't know. I'm not a specialist. I'm not a breeder. But maybe it could be interesting for niche species for which there is no genomic information, or very complicated, like, I don't know, polyploidy and other things. And also that's something we are really interested in, is multi-species predictions in the context of agroecology. For example, looking for species for a given ecosystemic service. Thank you for attention.
Thanks again for the speakers, and we have some time for microphone. Yeah. So thanks again to all the speakers, and we have some time for questions. Are there any questions from the audience? Yeah.
Question for you, Gibon, for last speaker. You said that part of the time the reader spent analyzing the data. So do you take this into account? Manually adjusting the data, that's how feelings. So did you did you account this time into your cost?
No, yeah, I was just looking for the production of the data. So the data platform.
But then what about looking at the chromatograms, looking if the integration was done right?
That was, yes, that was counted.
That was that, okay. And the second question is the difficulty to compare data produced by different platforms. Now, this has been achieved for GCMS, for instance. Calibrate, you put calibrators in your run, you have a very rich database from NIST, so you can compare data from different platforms.
Yeah.
Do you think this will be achieved sooner or later for LCMS?
Actually, when you mentioned GCMS, I think you mentioned targeted analysis with standards. So if you use the same standards, you may have some good production. But LCMS, the non-targeted LCMS is another dimension, I think. I think that it could be solved, but labs would need to use the very same sample or extract to distribute the same sample to all the labs, and this could be used to integrate the data with normalize it.
Thank you. This might sound as a stupid question, but listening to all these, we have a lot of data experts here in the room. And knowing that we are struggling with more the complex issues that you mentioned in talk. And I saw on one, I think only on one slide, I think it was James' slide, it said machine learning. To what extent can we count on the development of AI and machine learning to handle these huge, sometimes difficult data volumes? I think to me it sounds like a good an avenue to solve many of the problems. I would very much hear your views on that.
First thing, machine learning, artificial intelligence already knew the process that, especially when going from raw data to majors. Now for the most one of the most time-consuming part, which is annotating the data, taking the table, ensuring that everything is well described. So far, I haven't seen something super from we have just initiated a series of projects with technical projects things for explorating interoperability for those first steps, which is about acquiring the data and correctly describing it. I'm absolutely not aware of the huge breakthrough in what I just mentioned so far. If I hear about it, I can show you that I will jump on it.
Any Any further questions? Otherwise, I would also have a question to basically the data experts. So we put a lot of effort in making data fair, making this data accessible, reusable. Do you have any estimates about the rate or the reusability in reality? How many people are reusing this data? Is there any measure? Do you have any estimate? Because we are often asked, you need to make this data available. You put a lot of effort into doing this, and often when you talk to people, they say it costs me a lot of work and I don't see a benefit. But having some data about this would really help.
So there might be, this is an important overview. There might be some studies or review that we should do some review of this. Know the components line. But more seriously, if I follow some of the data sets I'm working with, I see that there are hundreds or even thousands of downloads for some of them. What exactly happened with the downloads, I don't know. And although we should have a way to trace whether the DOI for those datasets are seated beyond the initial article, of course, beyond the initial authors. Yeah, but a study to have. If anybody has experience on that.
I think probably one of the main we use is within the same community. So when it's within the same community, I try to show some example. We use this straightforward between communities and people who don't know each other. This happens. Personally, I've done some of that, but it's more difficult, and the metadata more often than not are not good enough to do that. But you need to strike into the material and methods, etc. So, but I'm not sure there are good tools for that. But data are reused, but how much?
I think looking at your faces, you already spent 2 days discussing with us, which we really appreciate. And I think we, if there are no further questions on data, then we would move on to this general wrap up, which I do not know exactly how we.
Go ahead.
Let's not.
Please feel free to also ask questions.
I can ask.
Yes, microphone. And you are,
Close to the mic.
you also this one.
Okay, so this is time for wrap up. So we try to combine some of the discussions we had and to provide what we think are the useful outcomes. So we think that there are several stakeholders here, and all of these stakeholders have key and crucial roles in the upcoming challenges, and we would like to emphasize that we are in the brink of having a major loss in agriculture biodiversity, and we have also the possibility to contribute to avoid it. So there are major risks. We all have heard about pests and climate changes. So we also understand there is some difficulties for the gene banks. There are these funding very short. There is this need to have more people devoted to have higher visibility, I would say. And there is this perception that the access or the conditions for access or what you get from that access needs to be improved. So what is the race research infrastructure will work on? What do you want to do? That's up to you, of course, but we decided, so we put this here as you want to secure access and easy access to genetic material and information. So we need to have fair access to these resources. And of course, that
reasonable funding, adequate funding is necessary, and the public and private partnerships are most welcome, although still needing some work. Okay.
And with the yellow.
Yeah, yes, okay, no problem. So we talk a lot about phenotyping, and the phenotype is a very important part. Is a key aspect for reusing the phenotypic data, but also for promoting the use of plant genetic resources. It's really important, and so we have to see that also quantitative phenotyping is really relevant. Throughput is still a challenge, and during the different talks, we discussed several phenotypes, but very often it was a very simple phenotype, so we have to go really to the more complex phenotype and to include the effect of the environment, and it's really a challenge. There is a wide range of tools that can address breeding programs and agricultural management and agroecology. We discussed a lot about seed phenotyping, and with different tools that allow to have an appreciation of to analyze the variability of the seeds. We have a structure, 3D structure, volume, color, and function, for instance, aging. Concerning the cost-effective phenotyping, we discussed the different available tools. I don't remember all the acronyms, but it's volatiles, NIRS, LIDAR, bronze, and so on. So maybe it's we have many things in our hand, and we have to consider them. And maybe the major difficulty to my eye now is to manage all the data and to share this data to be sure that it can be variable for improving sustainable agriculture.
So what I try to do is to summarize or basically spend a few thoughts on how emphasis and grace INRAE could work together. So we have this opportunity. We are two different communities. Often there is a lot of synergies that we can use. Basically, this utilizing the synergies would be between those communities would be really, really important, and we can convert it not only in meetings, discussions, but something very tangible like joint projects. There was funding was mentioned a couple of times, and this actually should be also a goal that we not just discuss, but also for something very concrete. Include the industry and many different stakeholders. They are complementary, so I'm pretty sure that there is a way to find joint projects. I think the goal is quite often to select appropriate material for using in reading programs. So there are several. On the one hand, to selecting the appropriate material, but also this phenotypic characterization. So passport data, this is quite often a gap in addition to all this bottleneck regulatory issues and characterization of plant genetic resources. Bottlenecks, Ignacio will talk a little bit more about bottlenecks, characterization. These bottlenecks in regulatory issues. So this characterization of plant genetic resources. So we talked about cost-effective phenotyping. So there is a lot of technologies that we can use. There is a lot of standardization. We talked about data quite a lot, and this effective use of synergies, basically avoid duplication. This is something that's on the one hand, that's a good argument towards your national ministries that you need to address. This is a good argument for the commission that you also will need to address, we as well, and it provides also a lot of synergies that we can convert into practical things between grace and emphasis. That's maybe a little my perspective, learning the demand from the respective communities. So I have to admit, there are some gaps in completely understanding how to utilize the joint interests in a best possible way. So I still think there is a need to interact, to learn from each other, to visit a gene bank and see how those practical things work in reality, and to really understand how we as a phenotyping community could implement certain tools in basically practical things, which, coming back to funding, helps also in this sense to have a very targeted approach. And access to phenotyping tools, data, this is something that was addressed. So we have those data on both sides, I think, and can use them in a very, very useful way. And as such, I think this is not the end. This is it should be the beginning of a conversation that we should lead and continue within the next years. And I hope, I'm pretty sure there will be some useful output of it. As such, then some regulatory issues.
Yes. Okay. So sorry. So as research and infrastructure, we need to comply with the legal framework, the national framework. As mentioned in this today, we have two main instruments. One is the Plant Treaty and the other is the CBD Nagoya Protocol. With the big difference, the Plant Treaty is some other multilateral system more suitable for us, and while the Nagoya Protocol is as a bilateral agreement. In 2016, at the COP15, no, it's not COP15, at the COP, an elephant entered into the room. That is the digital sequence information. So sequence, the genetic sequence data, and some developing country want to apply the ABS access and benefit sharing rule to sequence data. This has a domino effect from CBD effect FAO, WHO with this big framework, WIPO and UNCLOS. To make a long story very short, this would be something as the end of the world as we know it. If the worst scenario if we have the worst scenario, because in this way, our common and widely used practice, the open access to genetic sequence, will be seriously hampered. An important milestone was reached in 2002 at the COP15, and this is the main issue. Is a multilateral mechanism would be a better solution than a bilateral one. Tracking and tracing DSI, the genetic sequence, is not practical, and the open access to sequence data, all the omics data, should be safeguarded. Negotiation is still going on. In the next year, at the end of 2025, we have the GB11 and governing body 11 at FAO, and this will be in the agenda. We will discuss about DSI. We will discuss about enlarging the Annex 1 to all the PGRFA, not only to 64 species, and also to introduce mandatory payment for accessing access and benefit sharing. That's all.
Okay. Okay.
You want to say something?
Oh, we have one more slide of if you.
And you have a slide of
There's one more slide. So before we have Giovanni wrapping up this whole meeting, there's one last Slido if we manage to get that underway. Let's see, there it is, okay. And that's, again, go back to your open, tab and connect to Slido. And so this kind of feeds into all the discussions we've had and the ideas about collaboration and how Grace could work for you. So the question is, how can Grace or I leverage your activities? So for your own organization, for your own research, for your own operation. So give it a go. I will turn on the visibility here. Oh. Yeah, I see a lot of activity, that's good. Networking, breeding, phenotyping. Access is necessary, phenotyping again, better exploitation of your data. Helping breeding support, increase the critical mass, remove barriers, legal issues. Collaboration, of course, joint activities, I think that's going to be very important. Still people are typing. I think you can give more than one response. But it seems, well, definitely networking is one of the main responses. Phenotyping, obviously, it's a phenotyping workshop. So thank you very much for your input. I think we will try to use this also in formulating some approaches for the next steps. Giovanni.
So what can you say after today that like this? I think that the most useful feature of the workshop is if people come out of it with new or different ideas respect to when they came into it. And I think that to put together these 2 days, some of us came into this workshop thinking that, conservation and access to plant genetic resources is the main thing. And that's the necessary thing, it's the basis. But I think we all convinced ourselves now, we reached the consensus that that is not sufficient, that we need research. We have thousands, millions of genetic accessions. We have tens of different platforms for phenotype, and we eat phenotypes, we don't eat genotypes. And some of these platforms produce gigabytes of data and have a predictive value. And that's the value there. The value is that the fact that you can build a system to extract value for agriculture for the next century from all this extreme wealth of genetic diversity. And for doing that, let me repeat it once more, we need research. We need a research infrastructure. So that's why Grace is not going to be ECPGR on steroids, if I may repeat this assertion. Now, let me give you just an example. Today, we heard about the predictive value of phenotyping, et cetera, on the performance in the field of corn varieties, for instance. And think applying this not to a limited set of varieties, but to subsets chosen smartly of the genetic variation that's available there. And using predictive tools to apply these to the changing conditions, changing climate change, emergence of new pathogens. I think that would be invaluable for European agriculture. So, and I repeat, for doing this, we will need research. I'm not so much interested whether it will be grace or emphasis or some other infrastructure that will do it, or we will do it jointly, maybe. But the important thing is that we absolutely need this to adapt our agriculture to this century. And I think this is the message I wanted to give to everybody. Thank you all for being here. And I have a last thanks to the organizers of this. I think Nick and Daria, who's back there. And I think they did a great job in organizing these 2 days, and give them a hand.
So yeah, thank you very much, everyone. It was let's say a really good experience, and we have I think we look very positively back towards the fact that we brought so many different people together, etc. I still would like to thank also the online support from the ECPGR Secretariat, Nora and Valerio, and yeah, all the others that helped us. So thank you once again. This is only the beginning of something new. So looking forward to future exchanges, yes, I wish you all good travel back. And for those who were speakers today, we still have a present for you. If you did not, so please reach out to my colleague Daria before you rush back home. Thank you very much. Yeah, I wish you all the best. And
one last thanks, which I forgot. Our host Tony and Sig for hosting.
Yes, thank you, Sissy, for offering this meeting. Great. So thank you also for the online participants. Presentations will become available, also recordings, and we will make that in an easily accessible format online. So please check out the Grace INRAE website in the weeks to come. Thank you.